interesting about this beetle is that one of the haplotypes, namely the haplotype 1, shows a skewed sex ratio. So the haplotypes 2 and 3 are able to produce both male and female offspring, but the haplotype 1 for some reason doesn't and only produces daughters. And I want to know why that is. So the interesting thing is about these females that they still need to copulate with the males of the other two haplotypes in order to produce fertilized eggs. So the question that comes to mind is what actually happens to the male genes during the reproduction phase for the offspring to only be female. And that question I'm trying to answer using a protocol called Quadrat, which is a modified DD-RED protocol by Francini and co-workers from 2017. And I won't go into too much detail here, but the main gist of it is that you have your genomic DNA of your samples digested using 2 restriction enzymes. So one restriction enzyme is a frequent cutter and the other one is a rare cutter. And then you add specific base sequences to your digested DNA, and lastly you sequence it in one big pool, and then you will be able to call SNPs genome-wide simultaneously, as well as get rid of any PCR duplicates that will happen during the procedure. So I started off with 2 haplotype 1 families, a family meaning here that we have the mother beetle, the father beetle, and 5 offspring. In this case, because we're talking about haplotype 1, all the offspring that I looked at was female, of course. So in total, the program called just over 6,000 SNPs. And just to give you a general overview of what we did was we put the raw data into a pipeline of various programs. And in the end, we got something like this, which is a table with the locus ID, the SNP location, then the SNP of the mother and father, the SNPs of the 5 offspring, and then at the end, you can see what is called inheritance. This is the inheritance type that the program calculated, which isn't simple, it's quite a complex matter, but I won't explain that in detail now. So what we did analyzing this was we first looked at the SNP from the mother, so in this case a double T, then a double C for the father, and then went on to the offspring. So we have a TC combination in 2 of the 5 offspring, a TT combination in 2 offspring, and 1 missing data. So the program calculated that this SNP was inherited through sexual reproduction because part of the father allele ended up in the offspring. So this is basically what I'm now going to show you with the data that I have. So I looked at 2 haplotype 1 families, which you can see here. I know it's a lot, but on the left side, you can see the 2 families. The names don't matter right now. At the x-axis, you can see the 5 offspring each, and on the y-axis, you can see the percentage of inherited parental SNPs. What immediately draws the eye is that most of the SNPs, so up to 98%, were inherited either solely from the mother or through sexual reproduction. But what I want to look at is what happens to the male genes. So I'm focusing here on the right side. I zoomed it in a bit, so it's a bit easier to see, on the bluer schemed inheritance type. So I'm looking at the paternal gametes or sexual reproduction, or solely the sexual reproduction inheritance. Here I put the mean values of the 3 just mentioned inheritance types, so you can see with how low a percentage we're dealing with, and none of the inheritance types showed inheritance over 0.5%. So starting off, we have the sexual reproduction with a total of 83 SNPs that were called. All of those 83 SNPs had either a low or high likelihood, meaning that the protocol calculating the inheritance type was 100% sure that this SNP was inherited through sexual reproduction, or about 20 or 40% sure. In total, we have a mean value of 0.39% SNPs inherited through sexual reproduction. And this is what the SNPs looked like. Again, just as the example slide, we have a double T for the mother, double T for the father. 2 offspring showed a TC combination, 3 offspring showed a double T, just as the mother did. But as some paternal alleles ended up in the offspring, the program calculated this SNP as being inherited through sexual reproduction. Moving on, we have the paternal gametes. 19 SNPs were called in total for this inheritance type, with a low or medium likelihood, so not as secure as the sexual reproduction, meaning that a total of 0.09% of the SNPs were inherited solely from the father, and this is what it looks like. So I have a double T for the mother and a double G for the father. 2 offspring showed the double G combination from the father, and the rest showed the SNP from the mother. So we have something that hints at leakage of sperm DNA in the zygote during the fertilization, which we call paternal leakage, which is quite interesting, considering that all offspring in this haplotype is female. And because all offspring is female, it seems that there are complications in the meiosis of the mother beetle. So we still have one inheritance type left, which is the either paternal or sexual inheritance. For this, the program called 5 SNPs, all with a high likelihood, and a mean of 0.3%. These are the 5 SNPs that the program called, and again, a double A and an AT combination for the parents, and all 5 offspring showed an AT SNP, meaning that either they got the A from the mother and the T from the father, so through sexual reproduction, or the whole AT combination SNP from the father, meaning it would only be inherited through the paternal gametes. So the program calculated this must either be from paternal gametes or through sexual reproduction. But of course, 2 families aren't enough to really come to a conclusion. So I'm aiming at 10 haplotype 1 families to compare them to 10 haplotype 2 families. And right now, the data for 7 more families is waiting on the PC at university and is waiting for me to analyze them. As I just said and showed you, there is paternal leakage, or it seems to be that there's paternal leakage, and one possible reason for this could be the meiosis. And this is what I started during my first year as a PhD. Just to get general overview and to see how the cells look like in an unfertilized egg of a haplotype 1 female. I took DARPy stain pictures, which were sadly not as detailed as I'd hoped them to be. As you can see here, some of the cells seem to have problems dividing properly, as the chromosomes were condensed in weird places and not equally throughout the central lane, basically. So as those are not as detailed as I had hoped them to be, and I won't be able to say anything specific with these pictures, I'm moving on from this using antibodies. And this is something that I've just started. So we have 2 options here. First, I could stain the chromosomes using DARPy and label the microtubules using an antibody against alpha tubulin. But what I could also do, because this isn't optimal if I want to count the chromosomes, which I do want to do. So the first method that I will start off first is to label a centromere substructure called SID, which is a Sempier homologue in Drosophila melanogaster. And this substructure is present throughout the whole meiosis. So this labeling will make it possible to count the chromosomes at various developmental stages of the egg and see whether there's anything off with it, and then compare the results of the haplotype 1 counts with the haplotype 2 counts and see whether there's a difference between the two. What I think or what I expect is, since we only have female offspring in the haplotype 1 beetles, that there might not be a reduction of chromosomes in haplotype 1. The meiosis is basically made for the reduction from a diploid to a haploid state, but since there obviously is something off with something in the development of the haplotype 1 beetles, I'm guessing that it's due to the non-reduction of the chromosomes during the meiosis. So we would have diploid unfertilized eggs. But there could also be an issue with the fertilization due to the paternal leakage, because if it would solely be a diploid egg that would need the sperm to trigger its development, we would not have paternal leakage if the sperm wouldn't get into the egg. So after I check the chromosome counts, I will most likely go back to the microtubule labeling and see whether there's everything working fine, basically. So what is the general status quo so far? I have analyzed 2 haplotype 1 families, so all female families, and the data hints at paternal leakage during the fertilization of the egg and complications with meiosis.
Alina, you have, sorry to interrupt, 1 minute left of your presentation time.
Perfect, thank you. Which I will address in the next step of my PhD thesis. And ongoing, I have the analysis of 7 more families of both haplotypes still open. For the histological analysis, I'm aiming at 10 families for each haplotype, haplotype 1 and haplotype 3. But right now, as the pictures weren't clear enough or detailed enough, this is still quite open, and currently the beetles are hibernating, so this has to wait until the next year. So I would like to thank the whole hybrid consortium that I'm a part of, but most importantly, those who work with the beetle Altica lythri, and we wouldn't be anything without Altica lythri itself. And with that, I would like to thank you all for listening, and if you have questions, feel free to ask them, and I'll do my best to answer them.